with an intention to treat analysis, they took these two groups, which are the people randomized to medical, and then summed those up and looked at the groups. So you get 29, that's 27, plus 2 here. So that's intention to treat. And then intention to treat over here is the group randomized to surgical, whether or not they got surgical. So that's 21 who died. And so this is the intention to treat analysis, 7.8% versus 5.3%, a difference of 2.4%, which is not statistically significant. So in the intention to treat analysis, it's not significant. Then they did what they called a per protocol analysis. So they analyzed the people only in blue. Those are the people randomized to medical who actually got medical, the people randomized to surgical who actually got surgical. And in that case, you see a larger treatment effect that gets to be statistically significant, 4.3%. And then they did another kind of analysis, another kind of per protocol, and this is probably a big violation of ITT. It's called as treated. So they just basically ignored the randomization, and they looked at the people who actually, based on what intervention they actually got. So you group those who got the medical intervention, who were randomized to medical, with those who got the medical intervention who were randomized to surgical, the two blue groups. And that's called as treated because that's based on what they actually got. And again, now you see an even stronger treatment effect, 5.4%, which was statistically significant. So who's to say what the true value is of the surgery? But the point here is that you can get very different results depending on the type of analysis you do. Okay. So now you can do you can think of per protocol analysis as an analysis which is stratified based on a post-randomization variable because the adherence is post-randomization. There are all kinds of examples of this, and one thing again in the zoledronic acid trial, we were very interested to see, it's very tempting to want to do these kind of analyses because you really want the answer, it's just a very hard answer to get. For example, in the zoledronic acid trial, we wanted to ask the question, do those that have a larger increase in bone mineral density, are they better responders, and do they have a lower fracture risk? So it's a very interesting question, very clinically relevant question in a lot of cases. So you could think about stratifying people based on their first-year change in bone density and then looking at the fracture results. Well, that's based, the first-year change in BMD is a post-randomization variable. So our groups then would no longer be comparable. So even though it's a question we want to ask, and you'll see these papers all the time, and there probably is some value to them, you have to keep in mind it's a very, very biased kinds of questions because the people that responded in bone density in that first year are very different in the active group and the placebo group, and they're very different from those who don't respond. So more generally, any subgroup analysis based on post-randomization factors can be biased, sometimes extremely biased. And so you should be really careful and really wary about these kind of analyses. This is sort of the classic example of the effect of stratifying based on adherence and how they get you into trouble. This is a study of clofibrate versus placebo for 5 years. And if you look at just the one group result, it's 5-year mortality, you can see, oh, the people that took the drug who adhered had a lower mortality rate than those who did not adhere. So if you look at that, you say, well, what does that mean? It means, great, take the drug and it works, right? Well, no, that's not what you see, because if you look down here, you can see that, in fact, the effect of the drug was similar in the placebo and the treatment group, and the fact is that the placebo people who adhered did better than the placebo people who didn't adhere. So what's going on here is the idea here, the lessons from this, is that there are unmeasured confounders associated with compliance. In this case, for example, the younger people may have adhered better. Those who are more healthy may have adhered better, and that's why they did better. So it's very dangerous to do analyses broken down by adherence. Those unknown confounders may differ in the placebo and the active group. So again, the lesson here is to be wary of any analysis based on post-randomization variables, and that includes analysis broken down by adherence. Okay, so what are some of the problems with ITT or the full follow-up approach? Well, I think it's not the holy grail. Most importantly, if you're interested, is that an ITT approach doesn't really analyze the full biological efficacy of a drug or an intervention, because you're grouping people in the ITT who didn't take the intervention or didn't take it for the whole time. And so the utility of the drug may be underestimated, so you need to keep that in mind. And in particular, it's important for adverse effects. It may be an anti-conservative analysis for adverse effects. So I think the FDA generally will require that for the analysis of adverse effects, that a company that's going for approval of a drug include a per protocol analysis as well as an intention to treat analysis, because they believe a per protocol may be more conservative for looking at adverse effects. So again, the ITT is not the be-all end-all. So there are some cases where it doesn't apply. Now, the consort guidelines I wanted to mention because they relate to all of these things we've been talking about. You can look up the consort guidelines on the website here, and I think everybody likes it. The Lancet, the Annals, the JAMA, everybody likes it because it puts out guidelines for reporting trials, and there's a thing called the consort flowchart, which you won't be able to see this, but maybe on the printout or maybe if you're online. Basically, it tells you sort of how to lay out the patients. And I'll give you an example, again, from the zoledronate trial, which maybe is a little bit more legible. But basically, what you do, it's sort of a flowchart of what happened. How many people were screened, how many were excluded, why they were excluded, then randomized, then this is on one treatment. And in this particular case, these are going to vary a little bit by treatment. This, remember, was an annual treatment given over 3 years. So we talked in the active group, how many got 1 dose, 2 doses, or 3 doses. In this case, 2951 out of 3248 got all 3 doses. And then you can see the same thing in the placebo group. And then of those people who did not complete the follow-up, in this case 641, how many got how many doses and why they withdrew. This is a lot of detail, and this was negotiated with the journal when we were putting together the paper. They negotiated what they wanted in this, and so they kept saying they wanted more complete information. So the more information you have here, the better you can really see what's going on in a trial. And so this is really an implementation of the consort guidelines. And I think it's a very nice set of guidelines that really tells you how to report a trial. So right off, you can see here that in the efficacy analysis, there were 3,875 people. There were 3,889 that were randomized. So it's pretty much everybody. But there are 14 people that were excluded from the efficacy analysis. And why was that? Well, it turned out in this particular case, there was one clinical site in one of the Latin American countries that when they went out and monitored, they were concerned that there was fraud going on. And so halfway through the trial, they just stopped that study and decided at that point that none of that data would be analyzed because they had no confidence in it. So it's a valid reason, potentially could create some bias. But the point here is that the number here in the analysis, 3875, is, it's about 99-point-something% of the total randomized. And in some studies, you'll see that number maybe 50% of those randomized, and that's not good. And then in this case also, you can see the safety analysis was a little bit lower once again. And I think in this case, the company decided to also exclude people who never got the drug at all. And that was a predetermined decision. So whether that's right or not, I don't know, but it doesn't really make any difference in this case. What you can see from this particular case is that the follow-up was very complete. You can also look at how good the compliance was. This, I don't expect you to be able to read this, but this was from the Gaziano vitamin C and vitamin E for prostate cancer paper that we talked about last week in the first lecture. And again, they did the same kind of thing. They vary a little bit depending on what's included, but this is something that if you're really thinking about a trial and you really want to analyze it, there's a lot of useful information here about adherence, completeness of follow-up, et cetera. Okay. So now I really like the Hollis article, and they're Brits, and I have to say, we all know that the British invented our language, and they have the British have some things. That's British hot cuisine here, fish and chips, and there's the lady, and they definitely do talk better than we do.